Hello and good morning to UPSCFEVER.com. This week we are going to see Science chapter number 4, which is on cells. Right, so chapter number 4 is on cells. Um, every body, humans, plants, animals, everything in nature which is made up of tiny building blocks known as cells. And every cell has three basic components. One is the nucleus, one is the cytoplasm, and one is the membrane. So there are three basic components of any cell. The outer boundary of a cell is known as cell membrane, and this cell membrane is made up of fats and proteins. Most of the cells have a nucleus, and inside the cell membrane there is present cytoplasm. So this is the cell membrane, this is the cytoplasm, and this is the nucleus. So most of these cells, these three are the basic building blocks of a cell. Right, so these nucleus, cytoplasm and membrane are the basic components or building blocks of a cell. Next topic is the components of a cell. So these are the main components. First is endoplasmic reticulum. Endoplasmic reticulum is of two types. One is rough ER and other one is smooth ER. The rough ER has ribosomes attached to it, and ribosomes manufacture protein. And smooth ER has a important role in the manufacturing of fats or lipids. Endoplasmic reticulum serves as a transportation channel between various regions of the cytoplasm or as a transportation channel between cytoplasm and the nucleus. So in this diagram, you can see the rough endoplasmic reticulum and the smooth one. So this one has ribosomes attached to it, and transportation is one of the main functions of the endoplasmic reticulum. However, smooth endoplasmic reticulum also detoxifies poisons and drugs, and this is one of the functions of the smooth ER, but only in vertebrates. Next is the Golgi body. So you can see a Golgi body from this diagram. It is this structure. The main function of Golgi body is formation of an enzyme known as lysosomes. So you can note that lysosome is the enzyme manufactured here, and it is known as the suicide enzyme or the suicide bag, as it is used to digest wastes, digest foreign bodies, or even digest damaged cell organelles and the entire cell itself. The lysosome enzyme is responsible for digestion of the cell itself, and so it is known as a suicide enzyme, or Golgi bodies are also known as suicide bags. Another function of Golgi bodies is that it helps in making complex sugars from simple sugars and then storing these complex sugars in the vesicles. So the Golgi body has two roles. One is creation of lysosomes and creation of complex sugars. The next cell organelle is another very important one known as nucleus. We will reach mitochondria later. The nucleus contains chromatin material. You can see the nucleus here. The chromatin material is important during cell division as it gets organized into chromosomes. And there are 23 pairs of chromosomes in a cell. These chromosomes contain information which is inherited by the next generation in the form of DNA, and the functional unit of DNA are genes. DNA thus contain information which is necessary for constructing and organizing of cells, and any damage to this chromatin or the DNA or genes means a faulty copy of the next generation will be formed. Right, so nucleus is the key organelle of a cell. The next important organelle is vacuole. A single large vacuole, like you can see here, a vacuole is present here. This is an animal cell. So many small vacuoles are present in animal cells, but a single large vacuole is present in plant cells. The important function of this vacuole is storing liquid or solid contents. Then centrioles. Centrioles can be seen here. Yep. So centrioles are only a unique feature of animal cells, whereas similarly there is another unique feature found that is only for plant cells, and that is plastids. Green color plastids and white color plastids are the types of plastids. The green plastids carry an important pigment known as chlorophyll, and this chlorophyll is responsible for photosynthesis, whereas white plastids are called leucoplasts and they store starch, oil and proteins of the plants. Plastids have their own DNA and ribosomes. So you can see that plastids are found only in plant cells, whereas centrioles are seen only in animal cells. Membranes of the cell. Membranes, which is the cell wall, is a porous border and allows nutrients and other substances to move in and out of the cell. So this property is known as selective permeability. The exchange of gases from a cell is done by a process known as diffusion. Water is intaken or excreted by a cell by a process known as osmosis, and food is intaken by a cell by a process known as endocytosis. So these are the three important functions or processes by which food or water or gases are ingested or removed. An important difference between the plant and animal cell is that plants and bacterial cells have a cell wall which is made up of cellulose. And if you remember the first lecture, animals cannot digest cellulose. So the plant and animal cell does not have a cell wall. However, an important feature of this cell wall is that a plant cell can withstand much greater variations in the surrounding than animal cells. Presence of a rigid cell wall gives it the property that it can resist more changes in its surroundings. But there is a defect also, that certain antibiotics like penicillin, they block the formation of cell wall in bacteria, and that is how they kill that bacteria. However, since animal cells or human cells do not form a cell wall, penicillin does not affect us, but it affects the bacteria easily. That is a disadvantage of having a cell wall. Viruses lack cell membranes and they therefore do not show any sign of life until they enter a host. And this host can be either a human, or it can be a plant, or it can be an animal, or it can be a fungus or algae. The virus then uses the cell machinery of the host to multiply itself. Viruses have very few biochemical mechanisms of their own, and therefore it is very difficult to form antibiotics that affect the virus. Certain facts about cellular respiration are mentioned. In cellular respiration, oxygen is used to break down food and energy is released in this process, along with byproducts like carbon dioxide and water. So if oxygen is used in respiration, we call it aerobic respiration, And if respiration takes place in the absence of oxygen, then it is anaerobic respiration. Anaerobic respiration has an important use, that is in the making of bread and wine. Yeast is a fungi, and yeast has anaerobic respiration, and it releases carbon dioxide, and this carbon dioxide makes the bread rise. So yes, the second use of yeast is in the process of fermentation. Yeast is used to convert sugar into alcohol, and again, as we know that anaerobic respiration gives a byproduct of alcohol and carbon dioxide. So the alcohol is used in the process of wine and beer making. And this process of fermentation was discovered by the scientist Louis Pasteur. Right. So the next point is some miscellaneous topics are covered. Miscellaneous topics related to cells and organisms. As I have already told you, antibiotics don't work against viral infections, and penicillin, which is an important antibiotic, stops creation of a cell wall and thus kills the bacteria. But since humans do not have a cell wall, penicillin does not affect them. Analgesics are pain-relieving medicines. Antihistamines are relieving inflammation or used during allergic reactions. Similarly, antacid or antacids are also there. They neutralize the acid produced in the stomach and give us pain relief from acidity. Then important preservatives are sodium benzoate, sodium metabisulfite, and sorbic acid, edible oils, and propanoic acid. Though these are used as preservation of food. Sugar reduces the moisture content of food, and so it prevents microorganisms from growing on the food, and so sugar is also an important preservative. There are many types of sugar. One is aspartame. This is 100 times sweeter than natural sugar and is used in cold drinks. Then we have sucralose. This is 600 times sweeter than normal sugar. Saccharin is 550 times sweeter and is used to sweeten the food of diabetics. Since it cannot be metabolized, it is excreted in the urine. Aspartame is another artificial sweetener. One last point is about the process of evolution. So you might feel that evolution is, progressing into the next forms, but evolution does not mean progress from lower to higher forms, but evolution only is giving birth to newer complex designs even when the simpler designs continue to exist. So it is not that evolution always takes us from the lower steps to the higher steps, but it says that the complex designs will be formed, however the simple designs shall continue to flourish. And this was the question asked in the UPSC IAS exams preliminary 2013. Right, so with that the topic is over and there are certain questions. What are the basic components of a cell? So you should be able to solve all these questions. And if you solve all this, we can move to our next topic, next chapter, chapter number 5. So best of luck and see you. Thank you.